Oh, ladies and gentlemen, colleagues, friends, it's wonderful to see some faces from the past. From my past, not, I don't misinterpret yet. Right. So it's good to be back in Liverpool. We came in on the number 80 bus, and I've been away for 9 years now, so it really is a sort of lots of deja vu and things. Many wonderful memories of Liverpool. This is probably one of the best ones. There's Sir Simon, he's in the news at the minute, and there's me. This was the 50th anniversary of the Liverpool Mozart Orchestra in the Philharmonic Hall. That's some part of life I miss quite a lot, although I'm still playing in a piano trio. I'm now an emeritus professor. I'm trying to work out what that means. I know that edentulous means lacking teeth, so it's probably something like that. My wife, Geza, is a vet, and Hannah's here, and she has a younger brother called Sammy, and we're peasant farmers out here on the north coast of Antrim. And we look out over the Atlantic towards Islay and Jura. You can see 60 miles on a good day. And if you look carefully, you can see some black Welsh mountain sheep here. And they're the only registered flock in the whole of Ireland. And there's quite a bit of interest in them because they're very hardy. And people lost quite a few sheep about 3 years ago. So we're hoping to be able to sell those and at least not lose too much money. So if any of you are coming past this way, I can't imagine why, but if you are, you'll be more than welcome, and we'll feed you on best organic food. Right, so better cut to the quick, I think. I didn't realize when I put this title down for Max that it would actually be quite topical. As you know, up here in the northwest, there's been a report that fall in life expectancy for people at the age of 85. And there are a number of theories why this might be. One is a cut in funding for the NHS. But I think this is something we're going to see more frequently, and I'm going to tell you one set of reasons why I think that is. So there was a report in 2013 which asked where all the missing 90-year-olds were. I don't know whether you remember that. Basically, in the 2004 census, they would set a slope for the rise in average life expectancy. And they would predict the number of 90 and 100-year-olds that they would expect. And then when the next census came along, there were 30,000 90-year-olds missing. So clearly the slope had been put at the wrong angle. And here you can see these in the 90-year-olds for males and the over 100-year-olds for females. There were rather a lot missing than had been predicted. So these are all straws in the wind, that this inexorable rise in average life expectancy that we've seen over the past 100 years or more may be plateauing, and I think there are reasons why it might do more than that. Will it get worse? Well, we have an obesity epidemic, a diabetes mellitus epidemic, and a cancer epidemic. And the question is, are there common threads that can tie all these together? And there is one set of threads which hasn't had much. I mean, people who work in the field know about it, but certainly in the medical fraternity at large, I don't think it's been discussed all that much. And this is fetal programming. This is actually what can happen to you while you're in your mother's womb and how that can affect your pathology and future life prospects. And that's really what I'm going to discuss. We have to remember that the people who are making the going at the minute for average life expectancy, like my old man we're going to see tomorrow, he's 95 years old, they were all in their womb, the mother's womb, before what I'm going to tell you about happened. So the cohorts who are maximally exposed to some of the chemicals that are problematic have still got to come through yet, which is why I would predict that it is going to get worse. So we can have a very simple classification of pollutants which every one of us sitting in this room is carrying in their body. So persistent chemical pollutants are usually halogenated, organic compounds, they're fat-loving, and DDT is an example. If we took fat samples off people in this room, we'd be able to detect DDT or its metabolite DDE in most of us, if not all. And then there's a second set, because people began to realize that chlorinated chemicals had this problem of persistence and bioaccumulation. So then they started to make more easily metabolizable chemicals, and these include things like organophosphorus compounds, and we can metabolize those. And generally, within 72 hours, we can get rid of them from the body. But they tend to come in every day. And it's really the second group that I'm going to talk mostly about. We should realize that we're carrying a mixture of several hundred synthetic chemicals, and they weren't around 50 or 60 years ago. They're completely novel. And we have no way, as a toxicologist, I can analyze one at a time. If you ask me, I can maybe do two. But once you get beyond three, we don't really have the tools for analyzing the effects. And the highest dose is received at the most vulnerable time when you're forming yourself, when you're developing. So we are in the midst of a scientific revolution, and new research is changing the way we think about these things, pollution and safety and disease trends. And there are conceptual shifts going on. So classically trained toxicologists used to think there were a small number of chemicals that were a problem, but now we're realizing that many chemicals that we didn't previously think were a problem are actually causing problems. And these include those chemicals that can disrupt hormonal systems. We used to think that chemicals would act by completely overwhelming your defense system at high dose. But now we're finding that they can have effects, particularly in the developmental setting, in parts per trillion or low parts per billion. The sort of concentration at which your own hormones operate. And the old toxicology was concentrated on testing adults or adult animals, and now the new toxicology is looking very much more closely at developmental problems, and we find that the fetus is really the most at risk. So here's just a couple of examples. I mean, this was a big biomonitoring study done in the States, 700 and odd people looked at. They're looking for metabolites of organophosphorus pesticides in the urine, and you can see that, I mean, don't worry too much about the concentrations, but you can see that exposure is more or less universal. This was a set of organochlorine compounds in the circumpolar study, and again, you can see that there are differences between different countries, but all these chlorinated compounds here, DDE, DDT, Mirex, not only are they very complex, they have many, many thousands of congeners often, or enantiomers, but again, the exposure is universal. So now I just want to turn briefly to each of these three things that I mentioned at the beginning, because I think on the basis of just obesity, diabetes, and cancer, we can probably predict that life expectancy is likely to fall, from what I'm going to tell you in the next few minutes. I hardly need to rehearse these figures in this forum, because I'm sure all of you know that, that there are major problems with people being overweight. And this is the Global Burden of Disease study, showing that indeed over half of men and women now and a third of adolescents. It's a major, major problem. So I'm going to talk quite a bit about this study. I have a copy of it if anybody wants to have one. It's on this computer here, so you can take a copy. It was prepared by Miguel Porter, who's a professor of medicine in Barcelona and in the States as well, and Duck Hee Lee, who's a professor of medicine in South Korea. And what they've done is to review the science linking chemical exposure during the developmental period to the risk of obesity and diabetes. I don't know whether you'll be able to read that very well. It's a very crowded scale. But these are the classes of chemical which have all been associated with as obesogens. All right? So this is a term which was coined about 8 years ago. So these include dioxins and PCBs. These are all the persistent ones: organochlorine, pesticides, atrazine, perfluorinated chemicals, which are produced when you make Teflon, and polybrominated diphenyl ethers. These are flame retardants. That until quite recently, by law, they had to be in many fabrics and carpets. And then there are the short-lived ones, bisphenol A, which has been in the news a lot and is the one I'm going to talk most about, phthalates, which are softening agents for PVC, and polyphenols, which are foaming agents. And then there are a number of metals and pharmaceuticals and things. So in this report that I've just mentioned, there are 130 odd references, and anybody who wants to look further into it, the references are all here. So these are chemicals that have in vitro or in vivo, in animals or in humans, been associated with causing obesity. So they are defined as obesogens. And here is a study which was done that looked at a combination of 6 persistent organic pollutants, and they classified the level of exposure. And here you can see that the prevalence of diabetes associated with body mass index goes up to really quite a high level. It's interesting to note that this one, in the least exposed, even though they become obese, they didn't become susceptible to developing type 2 diabetes. So then we have a diabetes epidemic as well. 3.2 million are diagnosed in this country with diabetes mellitus and another 0.85 million undiagnosed. And we know that this is chewing up an enormous amount of NHS budget. These are environmental chemicals which have been associated with causing diabetes in these studies. And you'll see some of them are the same, dioxins and PCBs. Bisphenol A appears again, and a number of metals. So for this to work, it has to be exposed during the fetal period. Is there a common mechanism by which this might work? Well, what's being found now, and this is really, there's quite a lot being published on this at the minute, is that a number of the cytosolic nuclear receptors of the P450 system can be affected by these pollutants. And they can be affected at extremely low concentrations. As I say, high parts per trillion, low parts per billion. One of particular interest is this PPAR, peroxisome proliferation activated receptor gamma. This stimulates adipogenesis. So this actually controls the number of adipocytes you'll lay down in your body. Of course, they don't have fat in to start with, but they're sitting there. And if the fat comes along later on in the diet, then they will fill up. So it's a kind of pre-programmed obesity. Estrogen receptors alpha and beta increase adipocyte number as well. And DES and bisphenol A have been associated with that in research. PXR and this is the pregnane X receptor and the constitutive androstane receptor are xenosensor receptors, and quite recently it's being shown that they are also affected by lipid regulation. So all these receptors I mentioned have been shown in the scientific literature to be affected by chemicals that the mother passes across the placenta to the fetus. The aryl hydrocarbon receptor antagonists such as dioxin affect this PPAR and ER receptor by crosstalk. And finally, the thyroid receptor. BPA is a thyroid pathway antagonist. So if you think about the system, the ideal receptor occupancy is around 10% in a properly working system for most cases. If you can occupy some of these receptor sites with things that either stimulate or interfere, then these are very important in the developmental process. And then the third thing I mentioned was the cancer epidemic. The incidence of cancer has been going up inexorably for many decades now, and the most recent, and a number of those are hormone-related, so testicular cancer, breast cancer, prostate cancer. And it's also been going up in childhood. So all these factors are worth thinking about, but this was the latest paper that came out, which I'm sure many of you will have seen. But it's basically showing that the lifetime risk of cancer for people born since 1960 is going to be over 50%. And indeed, if you go into this paper, it's predicting later on that maybe 2/3 of people will develop cancer during their lifetime. When I was born, I think it was about 1 in 4 was the figure that was bandied around. So it's a big change. Okay, so I'm going to now talk in some detail about one chemical and just show you the effects that have been measured. And then we can try and extrapolate that to what I've just given us as sort of background. This is Anna Soto, Professor Anna Soto. She's a professor at Tufts University in Boston, but for the last 5 years she's been in our group for 20% of her time. George McCurr is a wonderful microscopist and was the guy who got me over there. Kurt Zetzler is a bioinformatician, and Stephen Downs is the head of the group, the head of the school. And this is our super resolution microscope, and it can resolve 50 nanometers with light. Amazing machine. So it just turns out that a number of chemicals that we've made, for whatever reason, are able to mimic the effects of naturally occurring hormones. This is nonylphenol. It's a foaming agent. Now, you look at it and you think, oh, yeah, that might be a candidate. We're not good enough just to look at it and be able to predict what it'll do yet. These structure-function relationships are quite subtle. You have to test it. This turns out to be an estrogen, and Anna Soto was the one who actually she was the first one to demonstrate that. That's a list of 49 pesticides which have all been recognized as estrogenic. You won't be able to read it, I don't think so. So I just want to emphasize the subtlety of this. Fred von Zaal is a scientist in America, a developmental biologist. And I should say that the people who are making the running in this now are not classically trained toxicologists. They are developmental biologists. He didn't do an experiment. He made some observations. And what he did was he looked at females incubating between 2 females in the rat uterus and compared them to females incubating between 2 brothers. And what he found was there was a predictable, lifelong change in behavior. And a number of other things. These young ladies had less estresses in a lifetime. But they were more aggressive. And apparently less sexually appetizing. I don't know how the hell they measure that. But anyway, and then he measured the natural estrogen levels, and these were exposed to about 100 parts per billion. And these to about These were exposed to about 100 parts per billion, and this young lady to about 130 parts per billion. So this minute change in concentration seemed to be able to cause a lifelong and predictable change in behavior. So we're dealing with rather sensitive systems. I think that's the main message from this slide. And what increasingly we've learned is that the environment interacts with genes to give consequences. That is the way the phenotype turns out. So toxicologists like dose-response curves. And this is what we like most, is a straight line. So up here in what I've called the pharmaceutical range of concentration, so parts per millions, parts per thousand, the sort of things you test drugs at. If you get a straight line here, you extrapolate back along it, and then you can say there's a no-effect level. And then when you have a no-effect level, you can set a regulatory limit for a pollutant. Now, what hasn't been happening is that people haven't been looking down here until quite recently. So I call this the cell signaling disruption range, parts per trillion, parts per billion. And when you look down here, all sorts of things are going on, and they're distinctly non-linear. You find these inverted U-shaped dose-response curves an awful lot, which of course is what you associate with hormone action normally. So what we're finding is that in many everyday products, there are chemicals which act as endocrine disruptors. So these are babies' feeding bottles. These are the linings of tin cans, and also in cosmetics. So this particular plastic is called polycarbonate. And polycarbonate is made from bisphenol A. Bisphenol A was discovered in 1930s, early 1930s, and it was developed as an estrogen. They were looking for a therapeutic synthetic estrogen that could be used in medicine. In 1938, DES, diethylstilbestrol, came along and trumped bisphenol A, and it was put back on the shelf and forgotten about. Within a few years, DES was being given to many, many pregnancies to prevent miscarriage. There wasn't a shred of evidence that it did. In fact, a meta-analysis since then has shown that it made it worse, but never mind. And this all came to ahead in something I'm sure most people here know about, is that some 20 years after DES had been given, so these are the DES sales, so translated 20 years ago, people started to find cases of a very rare vaginal cancer that had never been seen in a woman under the age of 65 before, in young girls of about 20. And a clinic might have 3 or 4 of these. So clearly people started saying, what is going on? And when someone put 2 and 2 together, the exposure history was in the mother's case notes. And this was a big shock, because prior to this, it had always been thought that the fetus was completely isolated from the environment, and that it was protected. But now it's clear that that's not the case. And in fact, we know that anything with a molecular weight of under 1,000 just sails across the placenta. So now I'm going to tell you about bisphenol A. So I've chosen a number of studies just to illustrate, firstly, this is multi-system effects. It doesn't just affect one thing, it affects many, many things. And lo and behold, they turn out to be related to what I was talking about earlier. So polycarbonate there's 1.5 million tons of this made globally each year, and it is made by the polymerization of bisphenol A. So bisphenol was rediscovered by the polymer chemists. They started making plastic they either didn't look or didn't realize that it's also an estrogen, but it is. Of course, whenever you start saying things like this, there's a reaction, and particularly the industries who are likely to be affected by any decisions regarding their products come back and start saying, but there isn't enough, there simply isn't enough there. Look at the amount of natural estrogen there is in a female patient, and this is just a smidgen. Well, of course, you have to think about these things a bit more carefully than that, because when a woman becomes pregnant, the steroid hormone binding globulin level goes up, and indeed the amount of free estrogen available to the fetus is quite low. It's carefully controlled. Whereas bisphenol A doesn't bind to this globulin. So it may be there at 1,000th of the concentration, but it's bioavailable. And that's the same with most of these synthetic xenoestrogens. They don't bind to the natural binding hormone. So here's a series of studies. So the first thing I want to tell you is that the so-called safe level of exposure of bisphenol A, determined by classical toxicology, is 50 micrograms per kilogram body weight per day. 50. And that is the regulatory standard as we talk. So all these effects I'm going to show you are at considerably lower levels than that. There's a big argument about whether this low-dose exposure has any effects. But, I mean, the science is pretty clear. So here is the effect of bisphenol A at 2.4 micrograms per kilogram body weight per day. So remember, this number 50 is the safe level, determined by adult toxicology on adult animals. And you can see that this induces obesity. This was first shown with DES. So this animal here was exposed by its mother to 1 part per billion of DES. Now, DES is a much more potent estrogen than bisphenol A, and this is the control. If you expose this to 100 parts per billion, it would be much lighter than that one. It's not a linear dose response. So this ability of estrogens to induce obesity is quite widely recognized now. So here's another one. This is prenatal exposure to bisphenol A in, again, mice. Reduces daily sperm production. It's significant by 20 micrograms per kilogram body weight per day, but it's already happening by 2. It's happening at a very low dose. This was in rats, same result. Reduced sperm count. Again, statistically significant by 20, but clearly something's happening before that. So this is the effect of prenatal diethylstilbestrol on adult prostate weight in mice. And I've put this in really just to show this nonlinear dose response curve, which we see very regularly in this field of research. If you give too much, it's lower than, statistically lower, and if you give just in this range here, where the receptor occupancy is clearly probably just right, then you get this massive increase. Again, by 2 micrograms per kilogram body weight per day here, an effect. So this is 1/25th of what is recognized as the regulatory safe limit. This is an in vitro study. It stimulates proliferation of human prostate cancer cells at low but not at high doses. So again, here's an inverted U. So if you give it up here, it doesn't have any effect at all, but if you give it in this range here, and these are the current human blood levels, if you do biomonitoring studies, but most people have some in them. And you can see this is in parts per trillion, quarter of a part per billion, very, very low dose. This is a behavioral study showing that when exposed to 10 micrograms per kilogram body weight per day of bisphenol A in the womb, then maternal behavior is disrupted. So this is the percentage of time outside the nest, percentage of time nursing. So these are all very subtle effects. They're not the sort of thing you're going to be able to just see when you see patients normally. I mean, it's and no one, of course, would suggest that any of these things that we're talking about is a single cause. They're all multi-causal. But I think these are very interesting findings. Now, here is a study. Here at 2 micrograms per kilogram body weight per day in male mice, you find an increase in aggressive behavior. This is testicular. As I say, I've just taken a series of these to show you. It decreases testicular interstitial fluid testosterone levels in rats, 2 micrograms per kilogram body weight per day. 1/25th of what we're told is safe. Now, it doesn't just affect mammals. Here at 1 part per billion, penis length in these marine snails is reduced significantly. Now, you may say, well, so what? I bet it bothers them. But this is increase in mortality of adult freshwater snails associated with malformations of the reproductive organ, 1 part per billion. And when we throw all this rubbish away, of course, it mainly goes to landfill, and then bisphenol A has been measured leaching from landfills. So the no-effect concentration for bisphenol A in freshwater snails is 8 parts per trillion. So this is what we mean by low-dose toxicology. So what do we have? We have effects that we can see in animals. So developmental abnormalities of genital tract, sperm production decrease, body weight increase, early puberty in females, I haven't covered that, hyperactivity and aggression, and there's also other changes in the central nervous system. Now, what we can't do, of course, is to map from animals to humans. All we can do is to note various things that are happening in the pattern of human pathology. I mean, there has been an increase in hyperspadias. I haven't shown that data, but I guess you all know that. Sperm count decrease, that's been widely trumpeted. Obesity increase, we've talked about that, and it's very clear. There's also a change to early sexual maturation in females. Well, I just want to finish by talking a little bit more about cancer now. Again, with respect to this low-dose exposure. So it's known that any child with a congenital malformation is marginally more likely to get cancer. We don't know exactly why that is, but it's a statistical fact. This is the work from Anna Soto's laboratory. So this is a flat mount of virgin mouse breast. This is the control. And this is the exposure to 0.025 micrograms per kilogram body weight per day of bisphenol A during pregnancy. Now, I don't need to tell you they're different. You can see. This has got more, longer tubule system, and we'll have a look at these end buds in a minute. This is 200,000 times below the current no effect level. So this is affecting the way in which this tissue develops. And they've gone on now to show that this is actually a pre-malignant condition. And indeed, more recently, they're now showing that it does lead to cancer. So I haven't got time to go into some of the theories that are knocking around about this, but teratology has been the study of malformations, of monstrosities, is the literal translation. But what's happening is we're causing teratogenic effects at the microscopic level. They're not obvious when you look at the clinical level, but if you start analyzing them down the microscope, you find these changes. And I'd look upon that as a form of malformation. You're getting dysgenic tissue. And Skarkebeck's work on the testicle, a dysgenic testis, has shown that that is a pre-malignant condition. So I think that this is, again, it's a straw in the wind, but I think that this is contributing to some of the changes that we're seeing in the epidemiology of human disease. So this is the testicular dysgenesis syndrome that I just mentioned. And I mean, what Skarkebeck did in Copenhagen was, if a patient came in with a seminoma, he would biopsy the other side. And he would find these pre-malignant changes in the other side. And his conclusion is that this is a developmental disease. And indeed, he went on to show that it was the hypofertile males who were more likely to get testicular cancer. So again, this is another sort of, so breast, testis, these are ones that we're seeing rising in incidence, and it seems that there is some fetal mechanism which could predispose to this. So I think with these compounds, precaution is really what we have in our armory. With the non-persistent chemicals, if you can get them out of the environment, then the problem will go away. The persistent ones are much more difficult because dioxins, for instance, have a half-life in the body of about 10 years. They're very, very difficult to metabolize and get rid of. I think it's time that strict liability was brought in for some of these industries that are producing some of these problem chemicals. And I think we need prior debate on a societal level before allowing some of these pervasive technologies to be developed. And these are all things that are being discussed at the minute. And the use of risk assessment has to be appropriate. It has to look at the most vulnerable period, and it hasn't done that so far. There is quite a lot of talk about getting rid of bisphenol A now. And it's certainly been banned from feeding bottles, where it was a major problem because people were using them over and over and over again, microwaving them, and it was shown that the stuff was leaching out. So I've got other stuff on persistent chemicals. I'm not going to talk about that because I've covered the main things that I want to say. So thank you very much.
Good. Well, thank you very much, Viv. It's fascinating, frightening, and also very controversial. I'm sure you'll be happy to take some questions.
Yes, yeah, if I can, I'll try my best. Yeah.
All research starts with imagination, I'm told. Certainly mine does, or has. You have to think that something might happen before you look to see if it does. And some of the things that you've shown us that did happen, I can't imagine how anybody thought, we'll look at that. We'll look at the length of the penis in a...
Well, I say once you got the initial idea, then you can start, because lots of people,
but how did you get it in the first place?
Lots of people work with very weird and wonderful animal models, and they think, oh, let's look at that. But yes, risk assessment goes through 4 stages, and the first one, which is hazard identification, is the most difficult. because once you've thought of it, then you can characterize the hazard, you can look at exposure, and you can do risk assessment. But when people started making many of these chemicals, why would they think about endocrine disruption? People who made organochlorine compounds, they could have predicted, if they sat down and thought about it, they could have predicted that they would be persistent. You know, in pharmacology, if you want to you've made a drug, but it gets metabolized in 2 minutes. The first trick in the book is stick a halogen atom on there and see if that helps. You know, so they would have known they were going to be lipophilic. They would have known, therefore, that they were going to magnify up the food chain. But nobody asked those questions because in those days you just made things and said, "Oh, it does that. Let's use it." So I think we have to really think very, very. I think in my lifetime, I've gone 180 degrees. When I was at school, I used to think new. New scientists had just been produced. You know, you go through every new. Oh, that's progress. That's fantastic. Now, when someone announces something new, my first thought is, what is that going to do? And try and do this hazard identification bit. And as many of you know, I did that with nanoparticles and caused something of a stir. But
well, thank you very much. Fascinating, if depressing stuff. May we show our thanks in the usual way.